Okay, so now we know that we're talking about protein synthesis. How do we make all these proteins in our cells? We know our blood carries proteins, but the proteins in the blood rarely leave the blood because they're so large. So what we're going to have is we're going to have our building blocks leave the blood. That's why we eat the proteins and break them down so we can get the building blocks, the amino acids. And we know the amino acids can enter the cells. Well, the next thing we've got to do is take these building blocks and actually build the proteins that our cells need. And there are literally thousands of proteins that our cells need to do the jobs of each individual cell. Okay, so how do we go from getting the amino acids that we got from our diet to building the proteins? How do we know how to put them together? How do we know what proteins we need? Well, we do this through this process called transcription and translation. Now notice here in our picture, we have our double membraned nucleus, and we have the contents of the nucleus, the nucleoplasm. The main thing in the nucleus actually being our DNA. We can say our double helix right here. Okay. What else we have is we have our cytoplasm, our contents in the cell but outside of the nucleus, and we see a couple structures here. The structure I want you to notice to begin with is a structure that's called a ribosome. Here we see the whole ribosome, and we notice that it's made up of two different subunits. Now, what else do we have here? Well, we see our three different types of RNA. Okay, first type of RNA we're going to be talking about is mRNA, also known as messenger RNA. So notice we have it here. Actually, this little area that I'm outlining right there is messenger RNA. This is messenger RNA, that's messenger RNA. Messenger RNA actually is made in the nucleus, leaves through a nuclear pore, and goes to a ribosome. All right, our ribosomes are actually made up of proteins and something called ribosomal RNA. So it's in order to have a ribosome, which is the factory where we're going to build our polypeptide leading to our protein, then we have to have ribosomal RNA because it makes up part of the subunits of the ribosome. Okay, the last extremely important type of RNA is tRNA, which stands for transfer RNA, because it transfers or picks up the amino acids and carries them to the factory where it then reads a code that was found on the messenger RNA. So notice that on the messenger RNA, which is this thing right here, we got these three letter boxes that are called codons. Now notice here it's GGG, which stands for guanine, 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 ACC, which stands for adenine, cytosine, cytosine, AAA, adenine, 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 GUA, guanine, uracil, adenine. And notice there's no thymine in here because remember, messenger RNA doesn't have thymine. It has uracil instead, but it does have adenine, guanine, and cytosine. Okay, now, so what we've got is we've got this transfer RNA bringing specific amino acids over. On one part of that transfer RNA, it has a code called the anticodon that actually is obligatory base pairing or complementary base pairing to the codon. So my transfer RNA has an anticodon. My messenger RNA has a codon. Notice how they are complementary to each other. Adenine is complementary to uracil. Cytosine and guanine are always complementary to each other. So everywhere I have an A or an adenine, I have a U or a uracil that has to match it. So if that was AAA, we know that it would be looking, if that was UUU, we know it would be looking for AAA. So notice that UUU is looking for AAA. Now, another thing about this that we need to understand is that UUU only picks up one amino acid. There are 20 different amino acids, but the transfer RNA with the anticodon of UUU will only pick up one. Which one do you think out of the 20 that is? Well, we have a little clue here because there's another transfer RNA with UUU on it, and it's actually picking up phenylalanine. Now, the anticodon UGG, guess which amino acid it can only pick up? Well, it only picks up threonine. Okay, so there's actually a chart that helps us figure this out, but I want you to notice from this picture what's going on. What that also means is that the codon AAA will only trigger the anticodon UUU, so it will only be specific for phenylalanine. The codon ACC, which triggers UGG because they are complementary to each other, it will also be a code for 3-read. Okay, so each of these is a code for a different amino acid, 20 different amino acids. We will go a little bit more into that on another quick explanation, but for right now, I want you to see this. Now, where did that messenger RNA come from? Well, what it did, it came from reading the DNA, and we're going to look at how it reads it in a minute. Now, to kind of back up and look at the bigger picture, what's going on inside of the nucleus where DNA is unwound at an area called a gene, and an enzyme is actually reading that and putting together this messenger RNA so it can carry the message of codons out to a ribosome to be read by the transfer RNA. All right, so we'll talk about how is it reading this. We'll talk about that on the next one. But this process that's occurring inside the nucleus is called transcription. Now, the process of the messenger RNA going and two ribosomal subunits come together, and then it reading the messenger RNA and the transfer RNA carrying an amino acid to the ribosome and building our polypeptide, which will become a protein. That process happening out in the cytoplasm is called translation. So again, we have our instructions here. We have our building blocks out here. We got to get our instructions to our factory so we can bring our raw products in and build our product. Forming the instructions happens in the nucleus, called transcription. Building the actual product at the ribosome is out in the cytoplasm of the cell. It's called translation. So protein synthesis made up of transcription and translation. Now let's look a little further for the next program at how this portion takes place, and then it'll help us understand how this takes place.